Assalamualaikum dear students. Okay, so today we are going to talk about cytokinins. Now, as you see, it's the name cytokinin. Cyto means cell and kinin means they cause mostly the cell division of the nucleus actually. So cytokinins are the hormones which actually promote cell division. Till now we were talking about auxins and gibberellins, and we saw both the auxins and gibberellins they promote cell elongation, but cytokinins are the ones that promote cell division. So and they are found in all tissues with considerable. They are found in all tissues. So these cytokinins they are found in all tissues with considerable cell division. Example, we will take embryo. The embryo is present in the seeds. Embryo is the one organ where there is active cell division taking place, and in this embryo you can find the cytokinins. Also you can find them in germinating seeds, because germinating seeds they also have this active cell division taking place. Young developing fruits also they have active cell division is taking place, and in these things, embryos, germinating seeds, and young developing fruits, you will find these cytokinins. So the roots they supply cytokinins upwards to the shoots, okay, and interact with auxins to influence differentiation of tissues, and they may be used to stimulate bud formation. So when we talk about this tissue culture, plant tissue culture, and we will see in plant tissue culture that these auxins and cytokinins they interact with one another to form callus, and from the callus, when a suitable ratio of cytokinin and auxin is applied in that callus, root formation, shoot formation takes place. So mainly these cytokinins are used for, like there is one example of cytokinin which is known as 2-BAP, benzyl aminopurine, and this benzyl aminopurine it is a cytokinin, and there is one more example that is kinetin. Both are used for the shoot formation, and for root formation auxins are used. So this is how they are used in this plant tissue culture or micropropagation techniques. Auxin and gibberellins they increase growth mainly by increasing cell elongation. That I already told you that auxin and gibberellins they promote cell elongation, but cell division is promoted by cytokinins. So growth involves another important process which is namely known as cell division, and this cell division is promoted by these cytokinins. So developing embryo shows active cell division, and that is why in developing embryo you will find large concentration of this cytokinins. Liquid endosperm of coconut called coconut water or milk. You'll find this is again a type of liquid endosperm, while this coconut water is actually a type of endosperm, and in which cell divisions are taking place. So these cell divisions are caused by a chemical known as kinetin. That kinetin is present in this coconut water or coconut milk. Similarly, the developing endosperm of maize contains factors such as zeatin. Zeatin is another, this cytokinin which is present in the endosperm of maize. As roots begin to grow actively in the spring, they produce large amounts of cytokinins, and that are transported to the shoot, where they cause the dormant buds to become active and expand. Now, when we see this spring comes up, so in spring what happens? The roots and shoots they grow. So firstly, the roots they grow actively and they produce this cytokinins, and these cytokinins are transported through xylem to the shoots, and in the shoot the dormant buds, okay, so they become active and they expand and form shoots like branches, flowers, fruits, and stem grows in this primary growth. So tissue culture is used cytokinins to induce shoot development. As I already told you that is tissue culture or micropropagation, they use cytokinins to induce shoot development, and they use auxins for the root development. So cytokinins may slow or prevent leaf senescence, leaf aging, or leaf fall. So in my next lecture I'll talk about ethylene and abscisic acid, and you will find that ethylene and abscisic acid are used for the promotion of leaf senescence or abscission or leaf aging or leaf fall. You can say they are all the same term: leaf senescence, leaf aging, or leaf fall. And here when we see that when we apply cytokinins to a plant, it is leaf senescence or leaf fall it may be prevented. Okay, so this is all about cytokinins. Tomorrow we are going to talk about these gibberellins, this ethylene and abscisic acid. Thank you.